My name is Griffin Rogers. I'm the director of the National Institute of Diabetes, Digestive, and Kidney Diseases, and I'm delighted to welcome you here today to hear Dr. Mitchell Lazar to tell us about nuclear receptors and the epigenomic regulation of circadian rhythms and metabolism. Dr. Lazar is a Sylvan Eisman Professor of Medicine and Genetics, the Chief of the Division of Endocrinology, Diabetes, and Metabolism, and the Director of the Institute for Diabetes, Obesity, and Metabolism at the University of Pennsylvania. Our guest speaker received his undergraduate degree in chemistry from the Massachusetts Institute of Technology. He then went on to receive his PhD in neuroscience and his MD, both from Stanford University. He trained in internal medicine at the Brigham and Women's Hospital in Boston and did an endocrinology fellowship at the Massachusetts General before joining the faculty of the University of Pennsylvania in 1989. Research conducted by Dr. Lazar in his lab, particularly the study of thyroid hormone receptors, it has led to really a series of discoveries related to the mechanism of nuclear receptor mediated repression. In addition, his lab has been using genome-wide methodologies to better understand the effects of diet, obesity, and circadian rhythms on metabolism, as you'll hear today. It's getting increasingly difficult to find leading scientific journals to which Dr. Lazar hasn't contributed. In fact, I was just looking over this, and just the first 3 months of this year, Dr. Lazar's work has been published in Science, Molecular Endocrinology, Diabetes, the Journal of Neuroscience, and the Journal of Immunology, just to name a few. Dr. Lazar has given named lectures throughout the world and is a former member of our institute, our NIDDK's council, our advisory council. He's also been associate editor of Diabetes and is on the editorial board of at least a half a dozen prestigious publications. In addition to NIH, many organizations have singled him out and recognized his seminal work and his outstanding achievement. These include the American Thyroid Association, the Endocrine Society, and the American Society for Clinical Investigation. I could go on and on about Dr. Lazar's accomplishments, but I suspect you, like me, would prefer to hear his presentation. Or with that, let me turn over the podium to Dr. Mitch Lazar.
Well, thanks, Griff, for that very flattering and great introduction. It's really a pleasure to be here, seeing old friends and colleagues, as well as meeting new people. And the NIDDK in particular and NIH are such spectacular places. It's really great to see the breadth and depth of research here. And although I'll only talk about, I'll try to be relatively focused, I'll start with some background that will hopefully hit on interests and areas of interest to a number of people in the crowd. But I thought I'd start with this because this reminds us of really what we are trying to understand. We're trying to understand what obesity, what causes obesity, and then how it causes its morbidity and mortality that it's associated with, and then what to do about those things. And one of the major things that we think about the morbidity associated with obesity can be boiled down to this notion of the metabolic syndrome. Jerry Reven, one of my mentors at Stanford, is the person who pioneered the concept of Syndrome X. And I think that from his point of view, and this is an interesting topic we could discuss, this is really insulin resistance, which otherwise is sort of one of the criteria that gets you into the syndrome of metabolic syndrome. He would, to this day, argue that this is an insulin resistance syndrome and that it's actually insulin resistance that's driving all the other bullet points on here. But as the metabolic syndrome has been defined, it includes this dysmetabolic parameters of central obesity, high blood pressure, high triglycerides, low HDL cholesterol, and insulin resistance, all of which are risk factors for cardiovascular disease. And of course, insulin resistance is sine qua non of type 2 diabetes. Type 2 diabetes also is epidemic, like obesity, and the leading cause of death from type 2 diabetes is cardiovascular disease, macrovascular complications associated with insulin resistance, as well as these other factors, whether or not they are caused by the insulin resistance or part of this constellation. What I'm largely going to focus on today, to keep a particular focus of the talk, is another aspect of the metabolic syndrome that's only, I would say, relatively recently recognized as more than just a curiosity, and that's the fatty liver. Not too long ago, although it was known that diabetics and obese patients had fatty livers, it was thought that it was really innocuous. But what is increasingly recognized is that the fatty liver is really a precursor of a severe problem. And the problem can be considered as on the slide, where the fatty liver can become inflamed and lead to so-called non-alcoholic steatohepatitis. That can lead to fibrosis, which can lead to cirrhosis. And indeed, fatty liver and liver disease associated with fatty liver is increasingly becoming a cause for concern, and the cirrhosis caused by it is one of the leading causes. I believe it's now second to alcoholic liver disease. Worldwide, of course, hepatitis C is up high on the list. And of course, cirrhosis is a precursor of hepatocellular carcinoma, and we now realize that obesity is a major risk factor for many types of cancer, and one of the leading ones is liver cancer. So the role of, or the effects of this dysmetabolic situation on the liver is of increasing concern. Now, if we're only this easy, if you go on the internet, you can actually buy this stuff. It's called Metabolic Syndrome Fighter. I'm happy to see that one of the exciting things that Dr. Collins is spearheading is interaction between the FDA and the NIH. And maybe the FDA will play a role in products like this. Now, at least it says it may assist in overcoming insulin resistance with an asterisk, so it's truth in advertising. But of course, if it were this easy, I wouldn't be telling you about this story. The other point I want to bring in as part of the introduction is the how increasingly clear it is that circadian rhythms play a role in metabolism, which we've known for a long time. All endocrinologists recognize that most endocrine systems obey a circadian pattern, but that actually they contribute to the dysmetabolism of metabolic syndrome and some of the issues our society is dealing with. Just to run through some of the many aspects of interrelationship between circadian rhythm and metabolism, many hormones have circadian rhythms. There are obvious links between circadian rhythms and metabolic physiology, in that whether you're nocturnal like a mouse or diurnal like a human, you tend to eat when you're awake, not when you're sleeping. Glucose and lipid metabolism both have circadian rhythms, and several cardiovascular risk factors, including thrombosis potential of the blood, have circadian rhythms such that most myocardial infarctions occur early in the morning. So there's sort of good news and bad news, as with any cyclic process. The good news is that we made it through the morning, and you have about a 75% lower risk of having a heart attack as you did it earlier in the morning, but tomorrow is another day. Obese people, of course, have sleep disorders, and sleep deprivation and shift work are associated with metabolic disturbances. And that's something that I think there were aspects of life, people that you knew or shift workers that you knew, sometimes nurses in hospitals, for example, who worked in the night shift. There was a sense that there were metabolic disturbances. But now this has been put in a more physiological context, largely by the large field of circadian biology. This is just a figure I've taken from the literature that tries to illustrate the concept of a misalignment between when the body would like to have its biological and chemical rhythms and how that's different from when it's sleeping and when the person is eating, and how this leads to this misalignment and maladaptation. And there have been a number of studies, particularly from Chuck Seisler and his colleagues, Shea et al. At the Brigham, which have, and many others, which really have made this clear in a controlled laboratory setting. but even observationally, there's more and more studies like this in the Occupational Environmental Medicine Journal, which I personally don't normally read very often, but you're starting to see more and more of these type of studies that make this association. These are people who were all on the day shift, and then some of them went to the night shift, and then this is the incidence of metabolic syndrome over the years as they either stayed on the day shift or switched to the night shift, and there's really a dramatic increase. One of the things which I won't talk about in the lab today, which we work on in my lab, and I think it actually may be one of my educational objectives, so I'll get it over with here, has to do with the relationship between the fat cell and insulin resistance. And one of the molecules potentially involved in this, which was discovered in my laboratory, is resistin. I won't go into detail on this other than to say that maybe tying this work in my lab together with that work is this observation that night shift workers actually have an increase in resistin, and resistin predisposes to insulin resistance. And now I want to show you an observation that was made almost 40 years ago that really doesn't have, has not had a molecular explanation. And I'd like to suggest that when I start showing the new work from our laboratory, that we have at least a plausible explanation for this phenomenon that's related to this relationship between circadian rhythm and metabolism. And here what you're looking at is mice, either wild-type mice or OB, genetically morbidly obese mice, which are on the upper part of the curve, of the graph here. And I'll really focus on the lower part, but they're really, the OB is an exaggeration. The fat mice are an exaggeration. And also embedded on this slide, and this is the article it comes from, is adipose tissue, which is similar to liver in this regard. But since I'm going to focus on liver, I'm just going to show you the liver data here. And what you can see is that when the mice, normal mice, are in this case are in the dark, so they're active in feeding, that's when they have their increase in fatty acid synthesis or lipogenesis. So de novo lipogenesis, fatty acid synthesis by the liver, has a circadian rhythm that we've known about for a long time, but the mechanism underlying this phenomenon has not been established, and I'm going to suggest one for you in this lecture. And then to introduce the players that I'd like to suggest are involved, which are the nuclear receptors, I want to just bring up the whole notion of whether this is related to genes or environment. And when you look at people like this, who probably have metabolic syndrome, but certainly are obese and are genetically identical, we actually know from the work of Mickey Stunkard at the University of Pennsylvania and many others, identical twin studies, twins reared apart, showing that there's a tremendous concordance in obesity as well as in type 2 diabetes in identical twins. So there's clearly a genetic component, and yet the obesity and diabetes epidemic that we are so aware of, that really became obvious in the 50s and have accelerated to the 70s, the 80s, the 90s, and show no sign of slowing down in this millennium. These epidemics occurred during a time when the environment changed dramatically. There was got so much easier to get fast food, high caloric food at low cost. You could drive to work rather than walk, and the lifestyles all changed. And yet this time frame is too short to for any meaningful genetic changes to explain the explosion of metabolic disease. So there has to be an environment component, yet we know there's a genetic component. And I think that I'm sure many in the audience are already thinking this way, but it's the science of epigenomics that, rather than having people get into fights at cocktail parties about whether it's the genes in the environment, I think epigenomics are what link the environment to gene expression. And here I'm talking about chromatin modifications, could be DNA methylation or histone modification, that result in changes in gene expression and function without a corresponding alteration in DNA sequence. Notice I'm not, in my definition of epigenomics, saying anything about heritability. In the epigenetics community, in the epigenetics world, because of the term, or the part of the term genetics, I think it's reasonable to consider, and as many do, require heritability, whether it's from mother to progeny or from mother cell to daughter cell, require heritability of the change. Epigenomics and the epigenome in this definition really refers to the way that the genome, which is more or less the same in every cell except for very specialized cells, of course, is configured in the nucleus. And that involves the nucleosomes with the histone octamers and these histone tails, which get modified. And it's really those modifications that I'll particularly focus on. And the reason that this is how the environment can interact in the genome is that things in the environment interact and impact and change the epigenome. And I'm dividing here the environment into the internal environment and the external environment. The internal environment in part as a homage to Claude Bernard, the famous French endocrinologist, and really first endocrinologist, who postulated the role of hormones and metabolites in regulating the internal milieu, the internal environment. At the same time, the external environment is particularly notable for the diet. Down up north where I come from, in Philadelphia, this is the great stuff that we love to eat and which is contributing to the problem. But of course, it also contributes nutrients and vitamins. You can't completely go without a diet. And the other environmental factor, which I already introduced, is the circadian rhythm and the sleep-wake cycle and so forth. And so nuclear receptors, which we've been studying, I've been studying since I was a postdoc with Bill Chin in the 80s and in my own lab at Penn for 22 years. There are many fantastic groups here at NIH who are also interested in nuclear receptors, can be thought of in one sense, and I'll propose to you that they are really transducers of environmental signals into changes in gene expression, yes, but through epigenomic change. So the model for nuclear receptors is that they are these transcription factors that bind to small molecules to regulate gene expression. And these small molecules are invariably derived from the environment. The internal environment and the hormones and metabolites is where the nuclear receptors, which were originally known as the nuclear hormone receptors, because the first modulators discovered were the thyroid hormones, steroid hormones, glucocorticoids, estrogen, sex steroids, and so forth, as well as metabolites, including, and more recently, metabolites such as fatty acids and oxysterols. And the external environment is also sensed by a subset of nuclear receptors. For example, the fat-soluble vitamins, vitamin A and vitamin D, get converted into nuclear receptor ligands. The retinoic acid receptor is the target of retinoids, and the vitamin D receptor, of course, of vitamin D. And there's a fascinating subject of xenobiotics. Frank Gonzalez here is really the leader, one of the leaders in this area, in which the nuclear receptors are, a subset of nuclear receptors are sensing in a very primitive immune defense way, if you will, drugs or so-called xenobiotics, non-natural compounds that are sensed by the nuclear receptors that then drive the transcription and synthesis of the P450 system in the liver that detoxifies these chemicals. So that's something I'm sure Frank has talked about here many times. Here it just makes the point in my argument that the family as a whole has potentially has in common this role of responding to the environment. So what do? Well, they regulate transcription, and they do so by binding to a whole set of genes. And I like to use the term systrome, partly because of the omics era, but also because this term that was coined by Miles Brown, it substitutes for saying all of the binding sites in the genome occupied by factor X. You could just say the systrome of factor X, so it's a useful term. Now, the nuclear receptors bind to these many sites in the genome, and I'll have more to say about that in a moment, and they recruit co-regulator molecules, and there are two flavors of these, lots of specifics, but the major ones are co-repressors that actually repress transcription in the absence of the ligand, or co-activators that activate transcription in the presence of the environmentally derived signal. And they do so by acting as platforms that deliver many enzymes locally to these regions of the genome. And this is actually an incomplete list, and I'd like to focus on the most famous ones, which are the histone deacetylases and the histone acetyltransferases that modulate the acetylation of lysine residues in the tails of the histone molecules making up the nucleosome and change the epigenomic structure in ways that eventually leads to regulation of gene expression. So that's the link that I'm referring to here. And what I'd like to do in the rest of the talk is to start focusing sort of more and more intensely, and I'm going to start with the histone deacetylases. Now, there's a whole family of histone deacetylases shown here. There's, I don't know how well you could see it, but class 1, class 2, class 3, and then the NAD-dependent molecules called the sirtuins, SIRT1, and so on, which are also becoming increasingly famous for their potential role in caloric restriction, longevity, and metabolism. Inhibitors of the non-sirtuin histone deacetylase inhibitors, though, really have formed the basis of so-called epigenetic therapy of a number of cancers. And I'm just showing you a list here. So in clinical use today are a number of molecules that have lots of other effects, and I really should emphasize that and make them up at the question and answer period. But what they have in common is that they inhibit histones and deacetylases, and especially the class 1 and often the class 2. And I'm going to focus on a particular class 1 histone deacetylase called HDAC3, and at this point posit to you that not all the HDACs are created equal. And I'm going to really focus on HDAC3 because we think it tells us a lot per se, and I hope you'll find the story interesting, but I also think it's paradigmatic for how we have to be thinking about molecules one by one, not just as this class, and again, most of these drugs are really doing a lot more than potentially what they would need to do for their therapeutic effect. So why do we focus on HDAC3? And the reason is because years ago, in the course of a series of studies in my lab and also around the world that were trying to understand how nuclear receptors work and led to the discovery of the corepressors, in particular NCoR and SMART, and then almost immediately, that was the era, many in the audience probably remember, when we were first starting to realize how important protein-protein interactions were, and the first thing people did was to take either pieces of these molecules or the whole molecule and look for potential interactions in things like yeast-2 hybrid screens, which were extremely valuable, led to some great insights, but also led to a lot of false positives, if you will. And we went very quickly, because these actually are very large molecules, about 2,500 amino acids each. Went from a short period of time where there was no clue what they did to having hundreds of molecules they could potentially interact with. And what Matt Gunther, when he was a graduate student in my laboratory, this is really our lab's version of slugging through how the corepressor interacts with the nuclear receptor through these corner peptides and then the complex, and that's what you'll find in this compendium of papers. But it was really Matt Gunther's work that I'm going to highlight now. And what Matt did was to purify the complex of SMART, one of these 2 corepressor proteins, from HeLa cells, the endogenous complex. And what he found was a very tight complex we refer to as the core complex that contains a couple of interesting molecules I'm not going to go into in detail, but contained a single histone deacetylase, and specifically histone deacetylase 3 in stoichiometric abundance. And we got interested in that because, as I've already mentioned, this is a regulator of epigenomic structure and potentially of gene expression. And the other thing that Matt discovered, which really surprised us, because we've been thinking all along, and I think it's largely true, that these docking proteins, the corepressors largely serve as docking proteins that deliver these enzymes and other interesting and important molecules to targeted sites in the genome. It turned out that actually HDAC3, the polypeptide HDAC3, did not have has intrinsic activity, but you could not see its activity if you purified it from E. coli or from insect cells or made it in reticulysate. You didn't see any enzyme activity unless you incubated it with the corepressor. And Matt was able to reduce this to a 90 amino acid segment that contains a so-called SANT motif, which cognoscenti in the audience probably know what it is, and for everybody else it doesn't matter. But it's a motif found in a lot of transcription factors, about 50 amino acids. It turns out it's necessary but not sufficient. There's another 35 or so amino acids. So this is about a 90 amino acid peptide, much smaller than the 2,500 amino acids in the corepressor. And this 90 amino acids is both necessary and sufficient, not only for binding HDAC3, but for imparting and for activating its catalytic activity. So in other words, when we found HDAC3 was HDAC1 was discovered, and then by in silico and other molecular technologies, HDAC1, 2, 3, et cetera, were found by because of their sequence homology. But if we purified HDAC3 in a classic way, we would have purified this complex because the HDAC3 polypeptide by itself really isn't active. So we took advantage of this by starting to study what happens if NCoR cannot interact with HDAC3. This is the work of notably two people in the lab, Takahiro Ishizuka, who did all of the in vitro biochemistry and cell culture work, and then Teresa Allengard, who courageously knocked in the mutation that Takahiro had found to abrogate the binding and activation of HDAC3 into C57 black 6 mice so we could understand the in vivo importance of this protein-protein partnership. So what Takahiro found was a point mutation in that SANT motif within this domain that we call the deacetylase activating domain is sufficient not only to block HDAC3 interaction, which I'm not even showing you on this slide, but actually now we're looking here at the histone deacetylase enzyme activity of an immunocomplex containing wild-type NCoR and this point mutant expressed at equal amounts in these experiments, not shown here. And you can see that there's almost baseline amounts of HDAC activity, suggesting that not only is this point mutation not binding HDAC3, but that HDAC3 is by far the most quantitatively important histone deacetylase in this complex. So Teresa went ahead and made mice. It was very important for us to do so because the NCoR null mutation is embryonically lethal, as shown by Jeff Rosenfeld and colleagues. And these mice in the C57 black 6 background were born in normal Mendelian ratios, and they looked completely normal. And what that told us was that since NCoR, as I just told you, is required for life, and so is HDAC3, as we've shown, and Scott Hebert and Eric Olson have shown, they're all both required. That says that it's complicated and that there's other partnerships. For example, we know that HDAC3 can interact with SMART, and we know that NCoR has other domains. And in this experiment, it must be that these other domains of NCoR that are conserved across species are what are critical for the survival. In the mutant mice, the NCoR is expressed at normal levels, but doesn't interact with HDAC3, and that afforded us, and Teresa in particular, the opportunity to ask the question, really for the first time, of is there a role of this corepressor complex, NCoR for sure, because this is a mutation at NCoR, but here it's even a more specific question. It's the NCoR HDAC3 interaction. And indeed, she found that there was an effect, and I'm going to just summarize very quickly the highlights at the descriptive level of Teresa's paper, which is shown here, so I can get into more new directions to try to explain some of the earlier phenomenology in circadian biology that I mentioned. Well, what she found in this experiment, where she took out the interaction specifically between NCoR and HDAC3, were changes in biological rhythms as well as glucose and lipid metabolism. The change in the biological rhythm was a change in the free-running period, the behavioral activity, that is the inherent circadian clock of these mice. And the reason that we even thought to look at that is because a couple of years before, Uli Schibler and his colleagues in Geneva had implicated Rev-erbá, and I don't know how well you can see it, but this is Rev-erbá here. This is taken from a review article by Joe Takahashi, that Rev-erbá is one of two, I'm not only showing one, negative limb in the circadian clock. And very briefly, the circadian clock, the transcriptional circadian clock, involves a positive limb led by BMAL1, which is a BHLH protein forming a heterodimer with clock and regulating in a positive way the gene expression of many genes that we think of as being regulated in a circadian manner in, you name the tissue, and especially in the suprachiasmatic nucleus of the hypothalamus. But one of the genes that they regulate is Rev-erbá, and Rev-erbá, being a repressor, binds to the BMAL promoter. That was something that Lei Yin in my lab had demonstrated. And Rev-erbá is perfect for this because it's a nuclear receptor that we've been studying for quite some time, and it lacks a C-terminus that, again, the nuclear receptor cognoscenti will know is critical for activation of transcription by ligand. Rev-erbá is lacking that. It's a constitutive repressor, but binds NCoR and HDAC3, this complex, and Lei Yin had shown that not only was NCoR required, HDAC3 required, but that the interaction was required in vitro and indeed it turned out to be the case in vivo as well. And I won't have time to go into it, but this whole process is stabilized by molecular heme functioning as a ligand for reverb, which has some implications, which I'd be happy to talk about from a metabolic point of view, but because of time, I'm not going to emphasize that here today. I want to just emphasize that the circadian phenotype of these mice was more or less explained by the defect in reverb because it could not interact with NCoR and HDAC3. The changes in glucose and lipid metabolism that we noticed, they were hyperketotic, they were actually protected from insulin resistance and hyperglycemia and a high-fat diet. That was a bit harder to explain. We originally focused on adipose tissue, partly because our laboratory is largely focused on adipose tissue, but when we did microarray studies and other studies, although they had less adipose tissue, we really found very little change in gene expression in the adipose tissue, whereas in liver, we saw some major changes that are again summarized here. I'm not going to go into them in detail. But really what I want to do is now focus on how is this occurring? What is HDAC3 doing in liver to affect hepatic metabolism? And there were a lot of potential targets. The reverbs, as I've just mentioned, because of this, and reverb has other targets, including involved in glucose and lipid metabolism, et cetera, but also all of the other nuclear receptors in principle. And we're particularly interested in the PPARs, LXR, thyroid hormone receptor, but any nuclear receptor that can interact with NCoR or SMART and HDAC3 is a potential here. And there are also a bunch of other transcription factors that have been implicated as potential interactors. So you can think of this as a problem of systems biology, because you have the system of this corepressor complex that can interact with many different transcription factors, just partially shown here. We have a phenotype that we're ascribing to the complex, and we want to deconvolute it and understand the contributions of individual components and interactors in the hope of understanding, for example, we do think that the circadian changes are largely through reverb, but maybe we can find that the other changes are through other factors and therefore imparts more specificity and understand the process better. So to go about this, what we decided to do, and this is largely the work of Dan Feng in the laboratory, a graduate student, is to determine the systrome of HDAC3 in the liver. Again, all the binding sites of HDAC3 in the liver. And this technology, I think, is extremely well known here. The work of Keiji Zhao with many histone-modifying enzymes. Gordon Hager, who's pioneered not only many of the nuclear receptor chip assays, but also hypersensitivity type assays and so on, which use similar technologies. But for those who have not seen this before, the beauty of this is you can look at the endogenous molecules in their endogenous setting with the natural genome of the tissue or cells that you're interested in. Here we're using the mouse liver. And the idea is that you chop up the DNA in one way or the other, you immunoprecipitate it with a specific antibody, and then what many laboratories do is interrogate very specific regions of the genome by something that can be called chromatin IP chip PCR, where either semi-quantitatively or quantitatively, using location-specific primers, you interrogate the gene of interest and you basically figure out if your factor was there or not. Well, with the advent of deep sequencing, we can now do what's called ChIP-seq. And in this case, instead of doing this biased analysis and looking at only the sites you're interested in, you can look genome-wide, and that's why this is potentially called cystromics. And this requires, cheap, deep sequencing, which is now possible, not as cheap as we like, but it's getting there, and really outstanding bioinformatics. And we've been fortunate to interact on this project with Shirley Liu at the Dana-Farber Cancer Institute and her colleagues, and in particular, Tao Liu, a postdoc in her group. And most of the bioinformatic analysis I'll show you was the work of Tao. Okay. Now, I debated including this because it's such a general audience and it's a detail, but I do think it's really important to point out how much, if you're going to get genome-wide data, that's great, but if it's bad genome-wide data, bad data is bad data. And it's very important to be certain before, especially because there's so much time and effort spent in analyzing these data, that you're looking at what you think you're looking at. And different ways to think about this, but one of the ways is to say that whatever the signal you're seeing that you want to ascribe to your factor binding shouldn't be there if your factor is not there. And so what we try to do as much as possible, and I'm showing you an example here, is to confirm the specificity of the CHIP antibody. Here what we've done, and this is the work of Zheng Sun, who I'll mention in a moment, we've made mice that have the HDAC3 allele floxed on both alleles. We can infect or inject these mice with adeno-associated virus, CRE, into their tail vein, it goes into their liver, and that takes out HDAC3 in their liver. We then look at the CHIP experiment here by CHIP PCR, for example, at the BMAL gene, and here it sites that we found by ChIP-seq, but now we're confirming we're truly due to HDAC3. You can see that the signal essentially goes away. These are just control, this is not, this got formatted wrong, but this is insulin, which is not expressed in the liver, there's no binding, but you can see that the signal goes away, and that reassures us that when I tell you there's a signal, it really is because of HDAC3. Okay, so what did Dan find? Well, she did her experiment, and the way the ChIP-seq works is that you get a whole bunch of reads of DNA, 36 to 40 base pairs, and here you can see, or bases, and here you can see she got about 28 million reads. And then these could be deconvoluted into peaks, and how you call your peaks is very, it depends on how you want to do your bioinformatics and how many false positives and false negatives you're willing to accept. But here, what we've done is we want one read per million reads, and the height of the peak has to be 9 times or more greater than that of the surrounding DNA. So fairly stringent. And even with that, we see about 15,000 peaks of binding of HDAC3. But one thing that we now always ask, and I would suggest people in the audience should always ask, is when did you do the experiment? So I'll tell you, Dan did this experiment at 5 p.m., which is also called ZT10. That's Zeitgeber time 10. ZT0 is when the lights go on in your mouse facility. So this is when the mice are generally not active and not eating. And we picked ZT10 for a reason, and that's because we knew that Rev-erbá, nuclear receptor, we know is recruiting this HDAC3, at least in the context of the circadian clock, is at its highest level at ZT10, and it has this really profound, this is the protein, but the RNA is very similar, has this really dynamic circadian rhythm, and you can see that it's almost gone at 5 a.m. or ZT22. And so, as originally considered in these experiments, the notion was, as I implied earlier, that we would know that, or imagine, and we try to figure out which of the sites of HDAC3 binding at this time point were due to Rev-erbá, and then by looking at 5 a.m. where there's no Rev-erbá around, we could infer that some of the sites we see here but not here were due to other factors, and by doing similar types of analyses, including knockdowns and so on, we could start ascribing peaks to matchups between the factor and the corepressor complex. And as I mentioned, other transcription factors possible as well. But the biggest surprise we got out of all of the results I'm going to show you in the remainder of my talk is that when Dan did this analysis at 5 a.m., when there's no Rev-erbá around, but we still expected to see all those other peaks associated with all those other sites, she basically found almost no specific binding of HDAC3 to the genome. When you looked at it in terms of what those sites were, and this is actually a bigger circle than it should be to scale so that we can all see it, they overlap, but again, it's 100 out of 15,000. Now, that's bioinformatic analysis. How can we be sure this is real? First of all, it's not because HDAC3 is gone at 5 a.m. There's plenty of HDAC3 there. So what Tao did was to put the data in the form of a heat map. What you're looking at here are all the sites where HDAC3 is bound to the genome at this time of day, all of these 15,000 sites. And what you're seeing is when it's a color of red or white, there's binding. So this is not surprising, but this is what contributed to this blue circle. And the question is, if we look at these same sites at 5 a.m., you can see that they're either gone or really attenuated quite a bit. If you look at it a different way and you say, okay, I just want to know what the average signal was at the center of these peaks, well, of course, it was very high at 5 p.m., but at 5 a.m., it was almost, it was much attenuated. Now, I think because of time, I'm going to skip this, but what this slide shows, and we can come back to it if people are interested, is that this rhythm of HDAC3 binding to the genome is maintained in constant darkness and also can be inverted if the mice are only provided with food during the time when they would normally be inactive. So I'm not going to take you through that, but that's the conclusion of these two slides. Even though it was surprising to us to see such a great attenuation of the signal at 5 a.m., the most obvious explanation was that reverb was more important than we ever imagined it would be for bringing HDAC3 to genes in the liver. To address this, we did the following. First, we looked bioinformatically under those 15,000 HDAC3 peaks, and we saw the half site, which we know is the nuclear receptor half site. Secondly, we performed ChIP-seq analysis for reverb, and what we found was a remarkable overlap such that about 90% of the HDAC3 binding sites overlapped right on top, and I'll show you in a minute an example of this, right on top of the reverb binding sites. And finally, using reverb knockout mice that we obtained from Bjorn Wenstrom in Stockholm, but then bred to 10 generations on black 6 mice to be comparable with all the other experiments. Basically, what we find, and what is summarized in this blue box here, but for all sorts of genes, including BMAL, is that we largely lose the rhythm that we see between 5 p.m. And 5 a.m., suggesting that indeed it is reverb that's recruiting HDAC3 to the majority of these sites. Now, what about NCoR? As I've set this up for you, NCoR was in between, and so it took us a while, but we finally got an antibody that could give us a chip that we believe for NCoR. And what Logan Everett, who's a postdoc in the lab, has done here is performed an analysis where we're looking on the y-axis of the signal of HDAC3 and on the x-axis the signal of NCoR. And what is apparent is that this is really very close to the unity line, suggesting that the binding of NCoR and of HDAC3 are really highly correlated. And the sites that we're looking at here are all of the reverb binding sites. And what Logan did here was to color code reverb binding by its strength of binding, giving you essentially a third dimension. And so these are the lowest strength sites. These are the highest in green, and then yellow and red are a little less well separated, but we can still see, I think, pretty clear separation there. And what this tells us is that there's a correlation between the binding strength of not only reverb and NCoR and HDAC3, but also of reverb. And I know you have some really outstanding modelers here. We do at Penn also, and so we're able to model this data. First I want to show you that these points in a way should be taken out because this is where the input was actually greater than the signal. But here's the sophisticated modeling analysis that we were able to perform, which I think speaks volumes. Okay. So what I've shown you so far is that unexpectedly HDAC3 really had a dramatically different recruitment to the genome at 5 p.m. where we saw it on 15,000 sites, and at 5 a.m. where we barely saw it on the genome at all. But now we want to know what it's doing. That's the other part, taking it from integrative biology to integrative physiology. We want to understand the physiological function of this. And to get at this, we first wanted to know a more biochemical question, and that is it causing histone deacetylation? And that's an important point because there's been some question about this. In fact, Keiji Zhao and colleagues here at the NIH have shown very elegantly that histone deacetylases counterintuitively are associated with actively expressed genes. And we actually see the same thing here. They're associated with genes that are on in the cell, but what we think we've uncovered is that they have to be on because otherwise they couldn't be modulated. Genes that are off continuously don't need to be modulated. And the way we can see that this is actually playing a role is by looking deeply through deep sequencing at histone acetylation. So what Dan did is to perform ChIP-seq for histone H3 lysine 9, and I don't think you can see it too well. You'll have to, I'm afraid, take my word for this. But at ZT22, when there's, remember that's 5 a.m., when there's very little reverb and HDAC3 there, so, and there's more red here at these HDAC3 sites, and there's less red here. So there's less histone acetylation at the time that HDAC3 is present, which fits this notion that HDAC3 is causing histone deacetylation. But it's just a correlation. It could be the other way around. It could be that having less histone acetylation, it recruits HDAC3. But thankfully, although cause and effect can be really tough in this particular type of analysis, here we have for our use the mice in which we take out HDAC3 in the liver. And when Zheng Sun does this and then provides the samples after a week to Dan Feng, and she looks, and this is just showing you we take out the HDAC3, and she looks at histone acetylation in the knockout at 5 p.m. Now it looks like 5 a.m., not 5 p.m. And I think I'll show you this. It's more dramatically shown here. It's a bit complicated. You have to look at the differences of these curves, but here's 5 p.m. and 5 a.m., this difference. But here's, you can see that at 5 p.m., but in the HDAC3 knockout, is even more histone acetylation than at 5 a.m. And what this tells us is, summarized here in cartoon form, is that at 5 p.m., when Rev-erbá is bringing NCoR and HDAC3 to these genes, there's less acetylation than at 5 a.m. when they're not there. Well, the notion would be that this is having an effect on transcription. The mediator of most of eukaryotic gene expression is RNA polymerase II, and I think you can see here that these genes that are bound by HDAC3 at 5 p.m., there's much less RNA polymerase at 5 p.m. And when HDAC3 and Rev-erbá are not that, there's more polymerase, and that's again summarized here. And then we went back to the mice in which we've knocked out HDAC3 in the liver, looked at their gene expression by microarray analysis, or transcriptomics, if you will, and looked at the genes that were either all the genes in the microarray, the genes that were downregulated, which wouldn't be by derepression, or the genes that are upregulated. And you can see it's only the genes that are upregulated that have this dramatic enrichment for binding sites by HDAC3. So that fills in this piece of the puzzle, and taken together, the conclusion is profound in a way, that it's the diurnal recruitment of HDAC3 that's on a daily basis causing remodeling of the epigenome and changes in gene expression. So just to show you what this looks like on one track in the UCSC browser, this is our favorite, the BMAL gene. This is Rev-erbá binding. So the height of the peaks here, there are probably multiple binding sites, but the height of the peak here is proportional to the binding strength, or at least it's the number of signals, chip signal we get. And you can see it's there at CT10, not at 5 a.m. Not a big surprise, because there's hardly any Rev-erbá in the cell at 5 a.m. But there's plenty of NCoR. I didn't show you that, but NCoR is not changed. And yet its recruitment is specific to the Rev-erbá site, and it's also only at 5 p.m. And HDAC3, which I did show you does not change in its amount and its abundance, is also, this is that dramatic recruitment I was talking about. So here's the BMAL gene, but this is happening at 15,000 sites in the genome. Here's what the histone acetylation looks like. It's somewhat complicated because there's presumably a clearing of a more nucleosome-free region here, but you can see the sides have more histone acetylation, and there's even more histone acetylation in the center, consistent with the notion that when HDAC3 is there, there's less histone acetylation. And the Pol2 being correlating with the lack of histone acetylation is actually very valuable, because you might think, despite everything I did to try to convince you that these were not artifactual data, that we're just not being able to do CHIP very well at 5 a.m. Maybe Dan's a graduate student, maybe she's just not as on her game at 5 a.m. like she was at 5 p.m. But you can see that it's the same samples, and it's another chip, it's another antibody, but it's basically a chip experiment. And you can see there's a signal here and not here. So that's not it. It's really a true biological result. Okay, so finally, as I alluded to, we want to know what the function of this all is. We're interested in the mechanism. We want to deconvolute this in terms of what factor is recruiting the corepressor and what the role of histone deacetylation is and so on, but what's the role in biology? And I've got to eventually link it to lipid metabolism because I started by telling you that we were going to explain the phenomenon of increased lipogenesis during the night in rodents. Well, the way we got at this was also by a bioinformatic approach. First, we looked at the genes just from the cystromics bound by both HDAC3 and Rev-erbá at 5 p.m., and you can see these were largely metabolic processes. But when you even add in another I would argue, much more stringent and biologically relevant criterion, that is that the gene is upregulated a week after we knock out HDAC3 in the liver, as well as having the binding site, lipid metabolism jumps to the top. So that was, we had a prepared mind for lipid metabolism phenotype, and that's what I'm going to show you. And we've looked at this in a couple of different ways. This is using a transgenic approach in which we mated crossed the mice that are flocks for HDAC3 with mice that express the Cre recombinase off the albumin promoter, which is liver specific, but begins in development and then continues through adulthood. And when you look at these mice, this is what they look like, and the really perceptive people can see a little bulge here. This is, I don't know how well it's projecting, but there's a larger liver and it's paler, and this is what it looks like when you take it out. And it was clear that loss of HDAC3 in the liver causes hepatomegaly and fatty liver. This is in 3-month-old mice fed normal chow. It's very important because the diet is normally a huge regulator of this, but here, even on normal chow, we see the fatty liver. But this could be a developmental process, et cetera, and that's where this model, again, comes in very handy, because we can do this in the adult. And when Zheng Sun does this experiment and takes out HDAC3, leaves the mice on normal chow, and just looks 2 weeks later, after control, they got a GFP, they got infected by an AAV, but it expressed GFP, versus the ones that had the knockout, you can see a really dramatic increase in the staining by oil red O staining, which stains for neutral lipid in the liver. This is just a section, but if you grind up the liver, look at total triglycerides, there's really a dramatic increase, 8 to 9-fold. For example, on a high-fat diet, C57 black 6 mice, a couple of months on a high-fat diet, they would have about a 2-fold increase in their hepatic triglycerides. So this is really a marked increase. And in collaboration with our stable isotope core and John Millar, we were able to demonstrate an increase in de novo lipogenesis in these livers, suggesting that HDAC3 and Rev-erbá, or HDAC3 for sure, is controlling this. And actually, we think Rev-erbá is important even in this aspect of the biology, because the Rev-erbá knockout also has a fatty liver, albeit to a somewhat lesser degree. And one of the reasons is potentially because there's a cousin of Rev-erbá alpha called Rev-erbá beta, which might be compensating in some way, where Anna Buga in the lab is currently testing this by looking at the Rev-erbá beta and the double knockout, but I don't have those results yet. So in conclusion, what I started by doing is to show you that we've known for a long time that there's a circadian rhythm of lipogenesis in the liver. This is just a sort of a computer version of the data I showed you, and also turned around from the way it was. But I want to emphasize that in the nighttime period for the mice, when they're active and feeding, they also have low levels of Rev-erbá, HDAC recruitment is low, and that's correlating with the lipogenesis. 12 hours later, Rev-erbá level's high, HDAC3 recruitment is high, it's recruited to the genome, and there's a marked reduction in histone acetylation. And I think potentially the most remarkable thing about this, other than that there's a very specific team of genes going to of proteins going to specific, albeit lots of sites in the genome, is that this is happening on a daily basis, day and night. And in fact, there is this there's a circadian physiology of metabolism that's keeping lipogenesis like this, and it's related to our feeding and fasting and so forth. And it's the role of, if you will, the loss of Rev-erbá that builds up the triglyceride in the liver, only to fall down again when the Rev-erbá comes up, goes to the genome, brings NCoR and HDAC3 there, and turns off lipogenic gene expression, as well as having effects on beta oxidation as well. And if we just look day and night, we only see, if you look at total lipid, it's more like this. This is lipogenesis, but the importance is really brought out in the experiments in which we knock out HDAC3, and you can't undo the synthesis, and the synthesis just continues unabated, and it really becomes dramatically increased, as I've shown you, which this sort of physiology being countermanded by the loss of HDAC3, albeit in an artificial setting, really shows you how devastating this could be in the long run. So in summary, HDAC3, this particular histone deacetylase, is rhythmically recruited to target genes in the liver by Rev-erbá, and that results in the circadian epigenome. The histone modification changes, and this modulates hepatic lipid metabolism. So the take-home messages to end. Every day, there are reversible changes in the cistrome, where Rev-erbá is binding, where HDAC3 is binding, and undoubtedly where lots of other factors are binding. And epigenomic changes, illustrated here by histone acetylation, that are occurring throughout the genome. There's not a specific clustering of these 15,000 sites, although it's highly specific. There are millions of potential sites. Although it's highly specific, this affects metabolism in a major way. The amount of lipid accumulation is not subtle. So what I would suggest is that in addition to the specific interest that we have into the understanding of this as a physiologic mechanism underlying circadian metabolism, trying to understand how it contributes to the pathophysiology of liver disease and fatty liver and so on, above that, this work, I think, serves as proof of concept that we can integrate these omics, cystromics, epigenomics, transcriptomics, and the sort of poor man's metabolomics, I showed you with the stable isotope work, in physiology and pathophysiology. I would call it integrative physiology, and actually use it to both explore and understand aspects of biology that we didn't know before. We didn't know what regulates the circadian rhythm of lipid metabolism in the liver, and this analysis, as complicated as it is, gave us this additional clue. So finally, I'd like to thank the people in my group that have performed this work. This is a fairly recent shot of the group, and the person who did the majority of the ChIP-seq work is Dan Feng, a really terrific graduate student in the laboratory. She had great help from Anna Buga, who did the work on the Rev-erbá knockout, and Zheng Sun, who developed the AAV-CRE method that I showed you that was so valuable for these studies and is proving even more valuable for other studies he's doing that I hope to tell you about in the near future. Shannon Mulliken, along with Teresa Allengott, developed those HDAC3 flox mice, and Teresa also developed and did the work on the NCoR HDAC3 interaction mutation mice that I just quickly went through as part of the introduction. A lot of the basic work we've done on Rev-erbá and its role in circadian biology was the work of Lei Yin, who now has her own, she's actually K99 ROO funded by the NIDDK and has her own laboratory as assistant professor at University of Michigan. I already mentioned this, but I can't emphasize enough how important the collaboration of Shirley Liu and Tao Liu was on the bioinformatics. And I want to thank Klaus Kessner and Jonathan Shug. Klaus is the director and a great colleague in his own right, but also director of our functional genomics core of our Penn Diabetes Center, which is NIDDK funded, and Jonathan Shug is the technical director. A lot of the infrastructure for the work is provided by the Nuclear Receptor Signaling Atlas. And all of the work I described was funded by the NIDDK, and celebrating its 60th year. I know there's a celebrations galore. Probably every day there's another party for the NIDDK here, and there should be because it's done so many great things. But for my own work in particular, none of this would be possible without the great support I've received from the NIDDK. And finally, for those of you who are taking the train to Philadelphia or beyond, take a look to your left and you'll see our brand new building. We're on the 12th floor. Actually, Francis Collins visited us yesterday evening at the opening ceremony for the new building. And so one thing he got to see, which I'm going to share with you, is that, and I don't think overhead was used for this, but they actually made a cake that was in the shape of the building. And our Institute for Diabetes, Obesity, and Metabolism is here. And finally, I want to show you, we just literally moved in about 10 days ago, and the day we moved in, it rained kind of like it did yesterday. And we look out at Center City, Philadelphia, and have a fantastic view, but 10 days ago, this was the view from our laboratories and actually from my office. And it reminded me of how lucky we are to be able to do science in the US, how important the NIH is, and how we were all inspired by many things, but including what I tried to start this work on, which is the importance of understanding the causes of obesity, diabetes, and getting to the next generation of treatments. And so I'll end with this fantastic report that was led by NADDK, Judy Fradkin, and many others. I played a small part that I think is a great blueprint for the future. So thanks again for the honor of giving this lecture, and I'd love to take any questions.
I think we have time for a few questions.
So, Mitch, that was a lovely talk. Would you comment on the role of HDAC3 in regulation of gluconeogenesis or regulation? Well, I'll leave it there.
Yeah, Mark Reitman asks a great question of, well, he asked specifically about gluconeogenesis, but I'll just say glucose metabolism. And one thing which we haven't published, and we just recently have these data, so I hope this is true, but I think it is that we really, because of the work of many outstanding laboratories, we thought this would be a great model of fatty liver-induced insulin resistance, and since one of the main things insulin does is regulate gluconeogenesis, we expected to see abnormalities in gluconeogenesis as well as frank insulin resistance, diabetes, and so on. But in fact, collaboration with Rex Heme, who directs our metabolic physiology phenotyping core, these mice have perfectly normal, not only glucose and hepatic gluconeogenesis, but they have normal glucose output, and they are not insulin resistant in the liver. So it's some sort of other pathway. It gets to this whole cart and horse, and this is certainly a model in which they're dissociated. There are maybe in the audience some people, certainly people in the world, who would not like them to be dissociated, but this is a case where they are.
Sure, you showed the great data with the with HDAC3 recruitment and K9 acetylation in the regular cycle, but I'm wondering how much do other chromatin marks play a role, and especially when you do the Cre injection, how much are those other marks affected?
It's a great question about other chromatin marks, and we have not looked genome-wide, and we're thinking about it. We're actually debating what, given that these things cost money and take time and analysis, exactly what the next things to do. One thing, largely because of the beautiful work from Gordon Hager, we might prioritize above that is looking at DNase hypersensitivity, and then go back and look at some of the marks associated with that.
Excellent work in the mouse model. So how could we translate it in human patients? Obviously, we could look at the fatty liver using MRI. And so is there anything we could do to simulate it in human patients and to see this relationship?
It's a great question. I guess it boils down to how do we know this translates to humans? And it's very difficult to know that. Even the question of, it's very clear that humans, although it's, I built the premise on the night shift workers, even that has not been extremely well studied. There, the people who've done that work have largely used hepatic enzymes, which by the time your hepatic enzymes go up, you not only have fatty liver, but you have steatohepatitis. They do go up in night shift workers, but the studies are poor and not well controlled. Someone really needs to do the study of fatty liver in night shift workers. And I know it's being planned, but to my knowledge, those data have not been reported.
Especially the epigenomic change in human.
Oh yeah, and then to ask whether this mechanism regulates it, it's very difficult. I don't have a non-invasive way to do that, and it's something we would love to do, and people have suggestions, but for now I think we have to use the mouse as a model.
As far as the alterance between 5 a.m. 5 p.m. is concerned, how much is due to the day darkness versus fasting feeding? So if you restrict food during night and vice versa, what is the prediction? What would happen?
Yeah, it's a great question. It gets to the chicken or egg thing about during the time that they're in the night, there also is when they're eating. So I rushed by it, but when we only feed them in the daytime, we completely shift the rhythm such that it's the opposite of what it was when they were voluntarily taking 75% of their food in the dark, which is what they do when they're left to free run on 12-hour light dark cycles. So you can interpret, you could say, well, that shows you it's really the food and not the clock. But the reason that's not so clear is because Uli Schubler has shown that the liver clock itself is entrained by food in preference to light. So we actually take that, and I think in general the circadian field would take that as a sign of it's the clock. But there are studies we're doing now, for example, fasting the mice. The problem there is the mice are not the same at the beginning and as the end of the, metabolically, at the beginning and end of the fast. But at least there we know there's no food, we're taking out food as a variable. So we're just waiting for those studies which get at it from another point.
Thank you.
I was just wondering, in the clinical trials from the patients who receive HDAC inhibitors, do you see alterations in lipid profiles?
Yeah, it's a great question and one we've been interested in. I can only tell you that if you do a PubMed search along the lines of your question of whether HDAC inhibitors that are given to cancer patients and others clinically cause hepatic steatosis, the answer is it's clear for valproic acid, which is one histone deacetylase inhibitor that happens to be given more to patients with neuropsychological problems than with cancer, but it's an HDAC inhibitor. They do get fatty liver. The other ones, it's less clear. But one thing I want to also emphasize that less from this work, but from a separate project which we haven't published that I didn't present here, but when we knock out HDAC3 in macrophages, we really can control things better than we can, let's say, in the liver. And we have an interesting phenotype. We do not see the same thing with HDAC inhibitors. And I think what that tells us is that HDAC inhibitors have off-target effects, and they affect other HDACs that might have opposite effects on these parameters. And so, on the one hand, one of the justifications for our work has been this helps us understand better what we're doing to patients we give HDAC inhibitors, but I think it really underscores how much better it would be if we specifically took out the HDAC that does what we want to do and not the ones that are causing untoward effects.
Exactly.
Have you checked whether there's a difference in the rate of release of free fatty acids from adipose tissue related to in these knockout mice, and so whether the fatty accumulation in the liver is purely due to de novo lipogenesis in the liver, or is it also because of increased supply of free fatty acids?
It's a good question. In the case of, it's sort of interesting, in the case of the NCoR HDAC3 mutation that I glossed over, but which is in every cell of the body, we did look at that and there was no difference. In these mice, because it was liver specific, we didn't focus on a primary effect on adipose tissue. However, we did notice alterations in, and there's no change in fat mass, but we did notice alterations in lipid oxidation, as there almost would have to be to get that level of accumulation. But we assume at the moment that those are cell autonomous in the liver. But we haven't formally ruled out some secondary effect on adipose.
I think Dr. Lazar has given us a lot to think about. Please join us next door at the library where you can meet him and have refreshments. Thanks again.
Oh, thanks, Griff.
Thanks for the plug.
Well, I don't want to